Good afternoon, my dear colleagues. I am very glad to see you again and to take part in our paleobotanical workshop. In my previous record in this February, I told about methods of the analysis of epidermis and epidermal characters which could be recognized in the compressions of Paleozoic plants. Now I would like to show, using the example of one locality, namely the Dubensky quarry, how these characters form concrete distinguishable types of the epidermal structure. The Dubensky quarry is located in the Orenburg region, about 100 kilometers to the southeast from the Orenburg city. Here is the locality. Gypsum is mined in this quarry since 1930s. They say that this gypsum is of very good, very high quality. The gypsum-bearing member is of the Congorian age. It belongs to the upper part of the Lower Permian. Plant remains were collected for the first time in the Dubensky quarry by the expeditionary force of the Laboratory of Arthropods of the Paleontological Institute of the Russian Academy of Sciences in 2019, and in 2021, the same expeditionary force revisited the quarry together with me. As a result, the nice collection of plant remains has been collected. Many of plants remains were represented as impressions and compressions. Some of them you can see here. Besides that, a lot of dispersed compressions was collected. Some bed planes, like this one, for example, are completely covered with such small pieces without any recognizable morphology. These specimens were treated with the method of bulk maceration. The large compressions seem good enough to macerate them and make slides for the light microscope. Unfortunately, most of them don't allow such a treatment. The lower and upper cuticles of the leaf do not separate from each other, and so cellular structures of the lower and upper epidermis are superimposed on each other and not available for detailed study. But fortunately, compressions from the Dubensky quarry fluoresce in the ultraviolet light and can be examined using the fluorescence microscope. Besides that, many small dispersed compressions appear to be naturally macerated. They are single-layer transparent and allow the examination in a light microscope without any chemical treatment. All this allowed us to distinguish several types of epidermal structures among plant remains from the Dubensky quarry. According to the relation to the morphology of plants and morphologically based systematics, compressions can be separated into three groups. The first group includes large compressions which associate with impressions and can be confidently assigned to morphological species. The second group includes dispersed compressions without any informative morphology, but with characteristic epidermal structure which has been previously described in some known species. So these compressions can be also identified within the normal morphological system. And at last, the third group include real dispersed compressions whose systematic position remains completely unknown. These epidermal structures should be probably described as new genera and species, but many of them are represented by a single specimen. So the material seems to be not sufficient for such description, and some additional material must be weighted. So let us consider successively these three groups. The first group, compressions associated with impressions. The value of the cuticles assigned to this group is that they expand our knowledge of the anatomy of plants known mostly by their morphology. And the first representative of this group is Dubensky cuneiform. This species includes such dichotomizing leaves with a trend to the pinnate division of the blade. This drawing shows such leaf. Due to this trend, it previously was assigned to Sigmophyllum, a genus which was characterized by prominent pinnate dichotomizing division of the leaf blade. Dubensky cuneiform takes a quite definite position in a row of variability leading from pinnate dichotomizing leaves of Sigmophyllum to palmate leaves of Lepidopsis type, probably reflecting the evolutionary transition to Chaetonialis. In the Dubensky quarry, Lepidopsis cuneiform is represented by impressions and compressions of ultimate and penultimate dichotomizing segments. Here are photos of these impressions, and these are drawings. Some of them resemble pinnacles of pinnate leaves, pinnate fronds, but the fact that they are present only at one side of, suppose, the rachis, like here, at one side we have pinnacles and at the other we have not. And this fact shows that they are rather ultimate segments of the dichotomizing leaves than pinnacles of the pinnate one. They are rather thick, and therefore these segments are rather thick and therefore often demonstrate numerous longitudinal wrinkles, as one can see in some dispersed compressions extracted from the bulk maceration. Sometimes they are so thick that their apices were compressed in the plane perpendicular to their longitudinal axis, forming such strange peltate remains like here. No epidermal structure of these leaves was known up to date, but the compressions of Dubensky cuneiform from Dubensky quarry fluoresced well, which made it possible to study the epidermal structure in detail. Both surfaces of leaf look the same. The costal and intercostal zones are well expressed, like here you can see. Costal cells are elongated and arranged in rows. All stomata are concentrated in the intercostal zones, forming thereby stomatal bands. However, this differentiation into costal and intercostal zones is not seen in some places. The cellular structure here is isogonal, and stomata are evenly distributed over the entire surface. I suppose that this presence or absence of the differentiation into coastal and intercoastal zones depends on the width of the segment and the closeness to its apex. No papilla are seen anywhere. Stomata are sunken and actinocytic. Here you can see them. Subsidiary cells are arranged radially around the stomatal pit. Often they are curved in one direction so that the whole stomatal apparatus looks like solar sign with spiral twisted rays. A very characteristic feature. You can see these curved subsidiary cells. Sometimes these cells are divided into two cells by tangential wall, so that the stomatal apparatus becomes incompletely dicyclic. Also, the majority of them are monocyclic. Stomatal pits are elongated and randomly oriented with a regular form conditioned apparently by the labiform thickenings of the proximal radial walls of the subsidiary cells. No guard cells were observed anywhere, and their shape remains unknown. The second species of the first group compressions associated with impressions is Dubensky insolita. It is represented by impressions of spiral twigs or isolated linear leaves. The leaves have dorsal furrows at their abaxial surfaces. It has grooves at the bottom of which all the stomata are concentrated. You can see these dorsal furrows here in translucent light and here in reflected light. The most astonishing feature of this plant is that its dorsal furrows can dichotomize in some cases. Dorsal furrows can never cross veins. And if we have a linear leaf or leaf with shape close to linear, its venation is usually parallel, like in this leaf of Rufloria, for example. And the multiplication of veins when moving from the base to the apex of the leaf occurs due to their dichotomy, like here. Veins dichotomize. Whereas new dorsal furrows located between veins appear through insertion, like here. This is a diagram shown by Sergei Viktorovich Main, which illustrates the emergence of new veins by dichotomy and dorsal furrows by insertion. Veins are shown by dotted lines and dorsal furrows by solid lines. But in Dubensky, we observe dichotomizing dorsal furrows. You can see them here. Some of dorsal furrows insert, like here, for example, or here, and some dichotomize. Veins were not visible in any specimen of Dubensky, and it was a puzzle how they behaved by such a pattern of dorsal furrows. This picture illustrates the epidermal structure of Dubensky. Green pictures are from the fluorescence microscope and brown ones from the light microscope. This is upper epidermis of Dubensky insolita. Cells arranged in regular rows. Neither papilla nor stomata are visible. The lower epidermis outside its dorsal furrows is identical to the upper one. The same regular rows of cells. These are dorsal furrows bordered by bordered at the edges by papilla, which are comparatively large. These are also dorsal furrows with different magnifications. This is just a place of the dichotomy of the dorsal furrow. You can see how it dichotomizes. And this is a dorsal furrow with a very high magnification. You can see that papilla are present not only at its edge, but also inside it. Dark spots on the bottom, these are probably stomata. This is one of such probable stoma. The guard cells are not sunken under the subsidiary cells, and the whole stomatal apparatus is due to that the whole stomatal apparatus is sunken to the bottom of the dorsal furrow. Guard cells are semicircular and bordered by papilla located at the subsidiary cells. Now the second group, dispersed compressions which can be assigned to known genera and species with known morphology. And the first species of this group is Pterophyllum gomankovi. This is a shoot of this plant reconstructed by Main and Smaller. It is conifer-like twig with rather long leaves with rounded apices. Unlike conifers, each leaf bears two dorsal furrows on its abaxial side. The epidermal structure of these plants is very peculiar. This is a general view. Where's my arrow? Oh. This is a general view of the lower leaf epidermis. Here is a dorsal furrow bordered by a fold along its right edge. Here is the same with a higher magnification. These big round formations within the dorsal furrow are stomata. The most peculiar feature of this plant is so-called double cutinization. We can see here, for example, two systems of cells which are superimposed to each other. Cells of one system have very thick radial walls and are arranged in rows which are not very prominent. Cells of the other system, on the contrary, have very thin walls. Which very thin walls and are arranged in a very regular straight rows. The reasons for this is that cutin was secreted not only by the upper periclinal walls of the epidermal cells, but also by their radial and even lower periclinal walls. So the radial walls seems to be very thick, and cutin secreted by the lower walls of epidermal cells penetrated even between hypodermal cells, forming the imprint of the second cell system on the cuticle, which correspond to hypodermis. One more peculiar feature of these leaves is that epidermal cells can be extended across the rows. You can see they are extended across the rows. Papilla, as a rule, are absent on the periclinal walls outside of dorsal furrows, but inside dorsal furrows they are very abundant. At the edges of dorsal furrows they are very big and can be named rather unicellular hairs than papilla. You can see them here. However, sometimes one can see rather large papilla on some areas on the epidermis. One of such areas located at the upper side of a leaf is shown here. You can see here rather large papilla. Such papillous areas with irregular location deliver one more very unusual feature of these plants. These pictures show a stoma at the bottom of the dorsal furrow with different focuses. The guard cells are hemicircular here and possess a cutinized roller along the stomatal fissure. From above, they are covered with large overhanging papilla. You can see large papilla which cover the whole guard cells. The second plant of the second group is Ensovia kungurica, a species described by Sergei Viktorovich Main from the Kungurion of the Permian region. These are drawings by Main showing linear leaves. The rounded apex is preserved in one of them. The characteristic feature of Ensomia are paired dorsal furrows on the lower side of leaves. They are arranged in pairs so that the distance between two furrows in a pair is much less than the distance between pairs. The cells on both sides of leaf form more or less prominent longitudinal rows. Most of periclinal walls bear papilla, which are more prominent on the cells adjacent to the dorsal furrows and less prominent apart from them. Along the edge of the furrow, papilla are rather big, resembling unicellular hairs. You can see them here, where the dorsal furrow itself is closed by a fold. Radial walls of cells often are finely convoluted or bead-shaped. You can see it here in some places and here with high magnification. These are stomata at the bottom of the dorsal furrow. They resemble those of Dubensky insolita. They are also circular, and their subsidiary cells bear papilla. Relatively small papilla near the contact with guard cells. The next plant of the second group is the Xydiella, a well-known conifer from the Congorian of the 4 Urals. These are drawings also by Main, showing leafy shoots which bear dense, spirally arranged cellate leaves with pointed apices. Several impressions of such shoots were found in the Dubensky quarry. You can see them here. But none of them in the Dubensky quarry retained any suitable cuticle, and only one compression of isolate leaf was found as a product of the bulk maceration. This one. Wait, excuse me. Xydiella is considered here as a member of the second group, dispersed cuticles with known affinity. These are views of that leaf at the fluorescence microscope. The preservation of the cuticle is not very good, but still one can see some details on the epidermal structure. The structure of both sides of the leaf is the same. Most of the cells are rectangular and arranged in the regular longitudinal rows. Many cells bear small papilla on the periclinal walls. And you can see this papilla in profile at the edge of the leaf here. Sunken monocyclic stomata are rare and poorly distinguishable. Seemingly, they are evenly distributed on the whole leaf whole leaf surface. The polygonal subsidiaries are usually less than non-stomatal cells and bear papilla which do not hang over the stomatal pit but are located in the centers of subsidiary cells. So this is the second group of compressions, and its value is that it expands our knowledge of geographical and stratigraphic occurrences of known plants. For example, Sterophyllum Gomankovi was known only from the Kazanian and Urrumian before it was found in the Dubensky quarry. And now its stratigraphic range is extended up to the Congorian. And now the third group, dispersed cuticles of plants with completely unknown gross morphology. The biggest group I consider. At least 7 different types of epidermal structure may be distinguished among these cuticles from the Dubensky quarry.
Alexey?
None of them was known before in any plants, and so they can be described as new. Of them are represented by a single specimen. Yes. Yeah.
It seems that we have some trouble.
Yes, my connection with internet was wrong. Do you hear me?
Yeah, now.
Now everything is all right now?
It seems so.
Okay.
Continue.
Yes. Okay, excuse me. Well,
We do not hear you now.
Well, yeah, 7 different types of epidermal structure may be distinguished among the cuticles from this third group. None of them was known before in any plants, so they can be described as new genera and species, but many of them are represented by a single specimen. Make me wait for more representative material for such a description. Due to this, I designate them as type 1, type 2, type 3, and so on. So type 1 is represented here. The cellular structure is isogonal, no differentiation of into costal and intercostal zones is visible. And although not very distinct rows of cells can be observed in some places, like here, for example. Stomata are randomly distributed. They are sunken and anomacetic. Idest subsidiary cells are similar in form to non-stomatal ones. The proximal radial walls of subsidiary cells often bear lip-like thickenings, misshaping the stomatal pits. This is stomata without preserved guard cells, and the irregular shape of stomatal pit is clearly visible here. The guard cells, when preserved, are very big, much more than stomatal pit. And you can see them here with different focus. And usually they are displaced relative to stomatal pit to one side. This is stomatal pit, and these are guard cells. They are semi-lunar in shape and often with strongly elongated polar ends. You can see them here. The second epidermal type. The differentiation into costal and intercostal zones is poorly expressed, but stomata are arranged in very regular rows and oriented strictly longitudinally. The radial walls of cells are poorly preserved, but non-stomatal cells look to be elongated, and it seems that stomata are sunken and anomacetic. The stomatal pits are seemingly very small and difficult to distinguish. The guard cells are fabiform. With strongly thickened polar ends and often with T extensions, like here, like T or anchor. Epidermal type 3. It is similar to previous type, but cells are less elongated, stomatal rows are less pronounced, and stomata are often oriented obliquely. In addition, most of the stomata are cyclocytic. The distal radial walls of subsidiary cells are thickened and form more or less regular round or oval contour. Here you can see such stomata without preserved guard cells and with very small stomatal pit. And here is a stoma with preserved guard cells. And you can see that, like in the previous type, they're fabiform with T extensions. Epidermal type 4. The cellular structure is isogonal. Cells are isometric, and any differentiation into costal and intercostal zones is not visible. Stomata look to be arranged in rows, which can be, however, more or less curved, like here, for example. Seemingly following the course of veins. Stomata are anomacetic and sunken relatively to the surface of subsidiary cells. But subsidiary cells themselves are strongly inclined towards the stomatal pit, forming an inverted cone-like constructions of the whole stomatal apparatus. These stomatal cones are very usually sloped to one side and pinned down to the inner surface of the cuticle during the fossilization. Here you can see one such stomatal cone from the inner side of the cuticle. The uppermost focus is on the guard cells. On the guard cells is here. Here are guard cells. Here it is on the here it is on the subsidiary cells. And the upper photo shows focus on the non-stomatal inner surface of non-stomatal cells.
Alexey?
Again?
It seems that we missed a sentence or two. You were moving your lips, but at least me, I didn't hear anything.
Oh,
just repeat two last sentences.
Do you hear me?
Yeah. Now it's okay.
Please, I have to repeat last sentence.
Yeah.
Okay. In these three photos, you can see one stoma with different focus. This focus is to the guard cells. This is to subsidiary cells, and this is to the inner surface of non-stomatal cells outside of stoma.
Thanks.
You can see that the guard cells are very small, no bigger than the stomatal pit at the stomatal, and the stomatal pit is regularly oval, has irregularly oval shape. The epidermal structure of this type is similar to that of Gingophyllum cuneiform in the shape of subsidiary cells, but difference in the absence of the differentiation into costal and intercostal zones, as well as in demonstrating stomatal rows and oval form of stomatal pits. Epidermal type 5 is represented only by unmacerated remains examined by means of fluorescence microscope. We can observe here the both sides of the compression of the leaf, and we can see that they look the same. The differentiation into costal and intercostal zones is absent. All non-stomatal cells look the same and are arranged in regular rows. Stomata are distributed randomly. They are sunken and anomacetic. Guard cells are not visible. Here is a stoma with a high magnification. The stomatal pit is polygonal and without any noticeable thickenings along the edge. Many non-stomatal cells bear papilla, but they are visible only with very high magnification. Here you can see them. But in small magnifications, they are not visible. Epidermal type 6 is also known from maceration compression, examined under a fluorescence microscope. For unknown reasons, the radial cell walls on one side of the compression are almost invisible. Here. The epidermal structure of the other side is isogonal. Cells are isometric and arranged without any order. Judging by the few areas on the first side where some cells could be distinguished, like here, for example, the epidermal structure of both sides was the same. Stomata are distributed and oriented randomly. They are sunken and anomacetic. The proximal radial walls of subsidiary cells bear lip-like thickenings, due to which the stomatal pits have an irregularly angular shape. Here you can see them. The guard cells are not visible. And at last, the seventh epidermal type. It is represented by cuticles where stomata do not present at all. Any stomata are absent. The epidermal structure is also isogonal, and cells are more or less isometric and arranged randomly. Sometimes the periclinal walls are supplied with papilla. You can see them here, many papilla on almost all cells. Strangely, these cells with papilla are concentrated in definite areas, whereas in other areas, like here, cells are devoid of papilla. It is possible that these cuticles did not belong to leaves, but to some other organs, maybe to seeds or something like this. Oh.
Well,
Excuse me, that's all I want to tell you. I tried to show a great diversity of epidermal structure observed even within one locality. Many of discussed epidermal types are completely new for science. Of course, the wide-scale epidermal analysis of fossil plants would expand our refine our knowledge of systematics of fossil plants, as well as that of their geographical and stratigraphic distribution. Thank you for your attention.
Thank you,
this
Alexey.
Strangely, it didn't appear.
Well, thank you for your interesting new results that you described for us. And anybody who want to pose questions may switch on your video, raise the hand, or alternatively write in the chat, as you prefer. And if anybody wants to pose the question in Russian, it's also okay. Well, meantime, I'll ask my first question. You told that guard cells did not preserve. Why can it be so?
Guard cells did not preserve in some cases. In some cases, they did preserve. I think it depends on the coutinization. In many cases, especially when guard cells are sunken under the subsidiary cells, the coutinization is very thin. Cuticle is maybe very thin and didn't preserve, or it may be totally absent. The guard cells were not coutinized. So in some cases, especially when they are sunken, they cannot be visible in preparations.
Well, in fact, I have one more question. All these plants from 3 groups, did they grow together, or they were transported to somewhere other place for burial?
I think they were transported from different places, but I'm not sure about the exact taphonomy, because I didn't examine the just a moment. I didn't see the real section, the real outcrop. All these remains were collected in the rocks which were excavated, already excavated from the quarry.
Mm-hmm. Thank you.
The staff of quarry didn't allow us to come into it. So we collected all plants here at the rocks which were already excavated.
Mm-hmm. Well, I see a question from Tatiana Alexeyeva in the chat. You may also read it in the chat. Thank you for the interesting materials. What is the most typical size range of epidermal cells?
The sizes are well about several tenths of microns. 30, 40, 50 microns.
Thank you.
In length or in width?
I'm sorry.
Length or width, several.
Usually cells are slightly elongated, so their length is more than width. And length is about 50 microns, for example, and width is about 30.
One more question from you.
I have one more little question about interesting structure like modern Ginga generative structure in one
Could you
slide.
show one of your first slides with gingophyllum or something?
Gingophyllum, yeah.
Just a moment.
It was somewhere at the beginning.
Oh, yes.
Yeah, this one. No,
the
previous.
Yes, this one.
It's maybe it's a generative structure or vegetative in right.
Lower right corner.
Yeah.
Do you mean this photo?
Yeah.
I supposed that it may be a generative structure, but no seed scars are visible here anywhere. And the epidermal structure of this remain is completely the same as in obviously vegetative fragments.
Very interesting.
So I suppose that it was the same, very similar segment of leaf which was compressed in other plane. Of course, I agree that this is very strange remain.
Yes.
I see one more question in the chat. The cuticles seem to be very nicely and quite well preserved. Did you try section by embedding them in resin? The question from Getan Gwinar.
No, I didn't try to cut them and examine in pen. Maybe it will be a future work.
Yeah, it seems that you have a scientist who does section in your lab. Natalia.
Yes.
Nosova.
Natalia Nosova does examines cuticles in sections, but I still not take part in these works. And I recently received this material, and it is the first stage of examination of it. So I only examined them in light and fluorescence microscope.
Gunnar added that details seem very beautiful for some of them. So it seems a promising idea for continuation of your work.
Yes, of course. I will continue. I shall continue these works in more more detailed methods. Maybe say so.
Thank you. Tatiana, you may switch on your phone to pose your question. Okay?
Yeah. Good afternoon, Alexey Vladimirovich. I have two questions. Did you publish a description of Dubensky already?
Yes, I have. It was published in Literarosica, number 26. Maybe I don't remember the exact number, but I think it was 26.
And the second questions about the dispersed cuticles. Do you have any ideas to what groups could they belong to? At least Spinalis, I don't know, maybe some of them, Litospermalis or others.
Most of them, for most of them, I have no such ideas. Maybe one type type 4 is similar to this Dingophilium cuneiform, and so to Platyspermalis. But for other, I have no idea.
Okay, thank you.
Thank you. There are two thanks in the chat from people who unfortunately had to run to other meetings. Well, do we have other questions to Alexey? Well, if we do not, then thank you, Alexey, again for your beautiful illustrated talk with very interesting results. Thank you all for joining us. Come next time.
Thank you for discussion and opportunity to talk, to tell us my results.
It was our pleasure. Well, see you all. And I see thanks for Ivan Gabrielian also.
Thank you.
The chat.
Bye.
Bye.
Thank you.